Cowabunga, brochachos.
Welcome back to Chapter 10. We are going to start today talking about photosynthesis. So we've just briefly mentioned it before, right? We talked about it with plants using energy from the sun in order to create carbohydrates. But we're going to talk about that process in depth. How does it happen? What are the different reactions involved? What pathways are going on? So we're going to start off by talking about photosynthesis in general. We already know use of sunlight to manufacture carbohydrates, photo and synthesis, photo meaning light. So we are using energy from the sun and we are making energy. We're converting energy from the sun into chemical energy that our plant can use. Now, autotrophs is what we define plants as, because auto means self and troph is referring to feeding or food. So when we say autotroph, we mean self-feeders, things that can make their own food. And plants do this through photosynthesis. Now, you and I, are heterotrophs. Hetero meaning different and troph meaning feeder once again. So these are organisms that have to get their energy, have to obtain their sugars from other organisms, from eating other things. So that would be like you and I. Now, photosynthesis as an equation is this right here. Okay, this little equation. And what we're doing is we're converting electromagnetic energy, which fancy word for sunlight, okay? We're converting energy from the sun into chemical energy. And you'll see our inputs are carbon dioxide, water, and light energy, so sunlight. And we are making, right here, a carbohydrate. Remember, this is our general formula for a carbohydrate. And oxygen as a byproduct. So if we look at this and compare it to Chapter 9, cellular respiration, it's basically the opposite, right? It's flipping cellular respiration. In cellular respiration, we're using a carb, we're using oxygen, and we're making carbon dioxide, water, and energy. So all in all, photosynthesis is just the flipped reaction for cellular respiration. And once again, this process of photosynthesis is a redox reaction. Okay, we are reducing carbon dioxide and we are oxidizing water. Now, we break down photosynthesis into two distinct sets of reactions. Okay, just like how we broke down cellular respiration into a few different sets of reactions, we're going to break photosynthesis down into two. So our first set is called the Calvin cycle, okay, and our second set are the light capturing reactions. Now, for the light capturing reactions, we also call this light dependent reactions because they depend on light. This is where we are going to harvest that energy from the sun and use it to create our high energy electron carriers. And you'll see our carriers are very similar to what we saw with cellular respiration. The only difference is we have an extra phosphate group if we're talking photosynthesis. So instead of NAD+, we have NADP+. But it's the same thing. It's an empty and a full electron carrier here. So in our light capturing reactions, we're using water. So this is where water comes in. Water is going to be split and oxygen is going to be formed as a byproduct. So this is where oxygen is produced. And once again, this is where we harvest that energy from the sun and we give it to these high energy electron carriers. Now, for the Calvin cycle, this is all about sugar. Okay, this is the set of reactions where we take some carbon dioxide and we make it into a carbohydrate. Now, for the Calvin cycle, this is also known as the light independent reactions because no light is involved in the Calvin cycle. Okay, all of our light we used in that light capturing reactions. So here's kind of a little overview. Here's our light capturing reactions right here. Green box, you'll notice capturing energy from the sun, using water, outputting oxygen, And you see we're making some energy and we're making some of those high energy electron carriers. Those are what's going to be passed on into the Calvin cycle, where they will be used to transform carbon dioxide into a sugar, into a carbohydrate. Now, where is this taking place? So with cellular respiration, the mitochondria was kind of our key location, right? It all took place in either the cytoplasm or the mitochondria. Well, for photosynthesis, the prime location is all about chloroplasts. We talked about them just briefly in our chapter on different organelles, but chloroplasts are unique to plants. And you'll see they're kind of similar in structure to mitochondria. We have this inner and outer membrane. On the inside, we have these kind of flattened sacs, which we call thylakoids. And when those stacks are kind of all stacked up and interconnected, we call those grana. The space inside the sacs is called the lumen, and any extra fluid, just kind of anything that's not a thylakoid, is called the stroma. So this would be like the mitochondrial matrix. Here we can see some different chloroplasts in the top. So these are our distinct plant cells. You'll see tons of little green chloroplasts floating around in there. If we zoom in on a chloroplast, this is what it looks like. We got that inner and outer membrane. We have the individual little sacs called thylakoids. If we see them in a nice little stack, that's called grana. Okay, stroma is just the space, the fluid surrounding. And that little blue sliver we're seeing, the inside of the sacs, those are the lumen of the thylakoids. Here is that looking at electron scanning, so we can see what it actually kind of looks like. Here we've zoomed in on a stack, and we can see, again, stroma, we can see grana, the stack, we can see individual thylakoids, and you can just kind of barely see that lumen inside the thylakoids. Now, before we can talk about photosynthesis, we need to understand pigments and what pigments are, because pigments are going to be very important to this process. Because if we look at our thylakoid, the membrane of our thylakoid, they have a ton of pigments. Pigments are molecules that absorb only very specific wavelengths of light, and they will either reflect or transmit others. So, just like how we see color, right? If you're looking at a blue Sharpie, you are seeing it as blue because it is absorbing every wavelength other than blue. It is reflecting blue. And that wavelength that it reflects is what you see. So pigments appear colored because of the wavelengths that are reflected, of the ones that are bounced back. Now, most common pigment that we're going to see in photosynthesis, it's going to do most of the heavy lifting, is chlorophyll. You've probably heard of it. It's been kind of a health craze recently, drinking your little green chlorophyll water. And I always say, if you're not a plant, if you're not going to photosynthesize, it's probably not going to do much for you. But this pigment reflects green light, which is why we see that nice, lovely, bright green color in a lot of plants and algae. It's because of the pigment chlorophyll. Now, a little recap on light, right? Electromagnetic radiation, it's a form of energy. And light can act as both a wave and a particle. Now, if we're talking about light as a wave, we kind of draw it out like this. Okay, it looks like a series of waves. Now, wavelength is the distance between the crests of a wave. So this distance right here is the wavelength. And this wavelength can tell us something about the energy of that light, because the shorter the wavelength, the more closely packed together those crests are, the higher energy the light. Okay, the further apart those wavelengths, the lower energy the light. Now, if we're talking about light as a particle, discrete little packets, we call those packets photons. Now, there is a whole host of wavelengths on the electromagnetic spectrum, and it really is a spectrum. However, we can only see a very specific section of light, which we call visible light. So this is just what is visible to us. Now, each photon and wavelength in that spectrum has a very specific amount of energy. And I already discussed it on the last slide, right? Inversely proportional to its wavelength. Okay, so the shorter the wavelength, the higher the energy. The longer the wavelength, the lower the energy. So here are all of the different wavelengths. So this is our electromagnetic spectrum, and you'll see gamma rays on one side, radio waves on the other. Now, gamma rays, X-rays, ultraviolet, these are all very high energy wavelengths. So very short little wavelength, very high energy. In fact, things like X-rays, so high energy that they can penetrate your tissues, hence why we use them for an X-ray. But if you've ever gotten an X-ray, can you see that X-ray when it is blasted at you? Can you see a gamma ray? No, okay, it is not in the visible spectrum. It is not a visible wavelength of light. Same thing for microwaves. If you've ever blasted a cold slice of pizza in your microwave, can you see the light? Can you see those microwaves that you are blasting it with? No, you cannot, because it does not exist in this little tiny range right here, which we call the visible spectrum. So this is our rainbow. This is our spectrum of visible light. Now, photons, remember those little discrete packets of light, can be either absorbed, transmitted, or reflected whenever they strike an object. And pigments, remember, only absorb certain wavelengths. And we can use a lot of really interesting techniques like thin layer chromatography to separate out different pigments from different plants and see kind of what wavelengths of light we would be able to absorb. What are we working with? So for example, here is a chromatography. Grind down some leaves, okay, put some of it on this chromatography paper, put it in some solvent, and you'll notice that the pigments begin to separate out. So we actually had multiple types of chlorophylls and some carotenoid pigments as well. So let's talk about our major classes of pigments. We already mentioned chlorophylls, right? Chlorophyll A and chlorophyll B. Okay, both chlorophylls, they do slightly different jobs, but we'll talk about that. These pigments typically absorb red and blue light, and they are going to reflect green light, aka that's why our plants are green, right? We are seeing that green wavelength that's being bounced back and hitting the light receptors in our eyeballs. Now, our second major type of pigment is carotenoids. These are going to absorb blue and green light, and they will reflect or transmit yellow, orange, and red. Now, when we're talking about pigments, we also have to understand a little something called an action spectrum. Okay, and what this means is certain wavelengths of light are better than others when it comes to photosynthesis. Okay, certain wavelengths are way better at driving those light capturing reactions, specifically pigments that absorb blue and red photons. Okay, so remember, chlorophylls, right? Chlorophylls are the main photosynthetic pigment because they are just better at it. Okay, that's a great wavelength for our light capturing reactions. And we can measure which pigments are better at these reactions by comparing our action spectrum to what's called an absorption spectrum. Okay, measure how the wavelength of photons influences the amount of light absorbed by a pigment. What that is saying is, for a specific wavelength, okay, how good is it at being absorbed by a specific pigment? So this graph here is kind of showing you exactly how the action spectrum and absorption spectrums are linked, how there is a strong correlation. So if we're looking at chlorophylls, right, that you'll see these peaks in our light absorbed, right? So the more light absorbed means the more oxygen produced, the better your reactions are being driven. Okay, so chlorophylls that absorb, again, this red and blue, they are going to have a higher amount of light absorbed, meaning they are better at driving photosynthesis than your carotenoids that absorb blue and green light instead. So you'll see with the carotenoids kind of this dip in our action spectrum. So they are not as effective as our chlorophylls at producing oxygen, at absorbing light. So what's the difference between chlorophyll A and B? They are pretty much going to look very similar, and they are going to absorb pretty much the same wavelength of lights. And generally, their structures have two major parts. So we have a long isoprenoid tail, which, remember, isoprenoid, we're talking about lipids. Isoprenoid was one of our lipids that we mentioned. And so this long tail is going to help interact with our cycloid membrane. And the head is this ring with a magnesium atom in the middle. Now, when we're talking light absorption, all of our light absorption happens in this head region. So here is A and B. Okay, here's the head region. Here's that magnesium. This is all the area that's going to absorb light. And again, that nice isoprenoid tail that's going to interact with our membrane. Now, the difference between A and B, pretty much a single functional group. If we're talking chlorophyll A, you'd have a CH3, and if we're talking chlorophyll B, we'd have a CHO. Okay, so pretty much the same molecule. So here is kind of a 3D rendering. You see that nice long isoprenoid tail and that head region with our magnesium right in the middle. Now, if chlorophylls are our main pigment, why do we even have carotenoids? What's the point, right? If they're not so good at driving photosynthesis, why do we still have carotenoids? Okay, and that's because they're called accessory pigments. And what these pigments can do is extend the range of wavelengths that can drive photosynthesis. Okay, so even though they are not as efficient as chlorophylls, right, we want to be able to capture as much light energy as possible, and that means capturing as many wavelengths as possible. So, increasing our efficiency using both types of pigments at once. Now, how carotenoids work is they are going to absorb that light energy, but they are going to pass that energy on to a neighboring chlorophyll molecule. Now, another interesting thing to think about is what happens to certain trees in the fall. We might not get a lot of it around here in El Paso, but if you've been to an area with a lot of trees, you might notice in the fall, right, those leaves begin to turn from green into red, orange, and yellow. And that's because when trees begin to die, their chlorophyll degrades, right? And when those leaves begin to die, when that green chlorophyll pigment is going to degrade, all that's left over is the carotenoids, those yellows, oranges, and reds. So here's an example of a carotenoid, so much different structure than we saw with our chlorophyll. And of course, structure equals function. So our carotenoids are going to absorb different wavelengths and they pass that energy on to our chlorophyll. Now, when chlorophyll absorbs a photon, so when that light energy hits that chlorophyll molecule, the photon's energy is going to essentially be transferred to some of the bonds in that head region of our chlorophyll. And if an electron is in that head region, our electron can become excited, okay, which we just mean when we say excited, a higher energy state. So it is an electron with a lot of energy, okay, it's excited. Now remember, chlorophylls can absorb red or blue photons, and blue photons are arguably the best because red photons, once they hit that head region, they will bump an electron up a singular energy level. So they will excite that electron a little bit. However, blue photons, when they hit that head of our chlorophyll, will bump that electron up 2 energy levels. Okay, that just means those electrons are going to be much higher energy than the ones that were excited by red photons. And remember, green photons are not absorbed by chlorophyll, they're reflected, and so they're not going to have any effect on energy levels. So here is showing you those excited electrons, right? A photon hits our chlorophyll molecule, and if it's blue, that electron gets really excited. It is bumped up 2 distinct energy levels, very high energy. If a red photon hits, that electron's only going to bump up 1 energy level, so not as energetic, not as excited. Now, whenever an electron is excited, so it's bumped up an energy level, just like we saw on the last slide, okay, when it falls back down to ground state, that energy that was absorbed, that bumped up our electron, is either going to be released as heat or light. Now, for talking light, that is what we call fluorescence. So, oftentimes, whenever that, once again, electron gets excited, when it falls back down and releases light, okay, we call that fluorescence. It'll glow. And typically about 2% of the photons that are absorbed produce some kind of fluorescence, and the other 98% is the energy we are going to use to drive photosynthesis. So this is where our photosystems come into play. Okay, photosystems are essentially a group of molecules, and it's going to be chlorophyll molecules that work together in order to harvest light. So this is all going to take place in the thylakoid membrane. Okay, we're going to see, again, 200 to 300 chlorophyll molecules and accessory pigments. And this whole grouping we call a photosystem. The pigments in our photosystem are going to kind of serve 2 functions. They're going to gather light. Okay, that's what we call antenna pigments. So they're going to gather up those photons. And that energy that they harvest from light, they are going to guide toward the center of this photosystem, which is called the reaction center. So what do antenna pigments do? Now, these pigments are directly responsible for absorbing photons. So they are gathering that light energy and they're using it to excite an electron, just like we showed before, right? Bumping that electron up in energy level. However, when we need to move that energy, okay, we are not going to pass that electron. It is not like an electron carrier. Okay, how we do it is by just letting that energy pass between neighboring chlorophyll molecules, but our electron always stays in the same place. So this process is called resonance energy transfer. We are moving that energy between chlorophyll molecules. So this is all going to be organized by proteins. They're going to help move this energy between our pigments, resonance energy, and the potential energy is going to drop each time that energy is moved. Once we've transferred that energy, that electron that we excited up, right, is going to fall back to ground state. And during that process, that energy that's released is passed on to the next chlorophyll molecule. So what can we do with that energy? That energy that's being passed or being released when an electron falls from an excited state back down to ground state. So there's kind of 4 options. Okay, we can either emit that energy as light, right? So it can just go be fluorescence. It can just be released. It can be given off as heat, so it can be released as heat or light. It can resonance energy transfer, right? So instead of just being released, it can get passed to the next chlorophyll molecule, whose electron gets excited, so on and so forth. Okay, just like that. Or it can be transferred to an electron acceptor in a redox reaction. So that means we could take that electron and we could give it to our empty electron carrier, right? NADP+. And once we donate that electron to NADP+, we are going to have NADPH. So here is our kind of little diagram showing the different fates for that energy. So those kind of four things that we outlined previously, right? So we excite our electron in the middle of that chlorophyll molecule. It gets excited up in energy level right up here. And when it comes back down, it's released as either light or heat, or we can do resonance energy transfer. So when that electron is excited up, okay, once it goes back down, it transfers that energy to a neighboring chlorophyll molecule, and that one excites an electron. And as it goes back down, it transfers that energy to the next, and it'll just play that little game of hot potato until that energy reaches the reaction center of this photosystem. Lastly, our last option is this reduction oxidation, right? So that photon hits, it excites that electron, and it is given to an electron acceptor like NADP+, right? So that's our empty electron carrier. And once that electron is given to it, we say it is now NADPH, okay, that full electron carrier. Now, one photosystem is great and all, but what if we had more than one photosystem, okay, working at the same time, working together? And that is called the enhancement effect. Okay, we can use two distinct different photosystems, each absorbing kind of different wavelengths, and we can use them to enhance each other. Okay, we can increase efficiency. And that's what we discovered was happening, okay? And we named those two different photosystems, photosystem 1 and photosystem 2. Now I'm going to ask, which one of these do you think came first? Which one of them is the first photosystem? If you answered photosystem 1, you are incorrect. Yes, photosystem 2 actually happens first, and then photosystem 1. Yes, it's stupid. I do agree. Okay, the reason they're named 1 and 2 is because of the order of their discovery. Okay, photosystem 1 was discovered first, photosystem 2 was discovered second. So here's a little graph of that enhancement effect. So you're seeing, if we're just looking at one photosystem at a time, we only get kind of certain wavelengths of red light in this case. But if we have two photosystems operating at the same time, we get this little enhancement effect. So not only are we getting those two wavelengths, we get a little bit extra because we got both operating at the same time. So let's talk first about photosystem 2. Okay, and we're talking about it first because it happens first in this little cycle. Once again, I know, kind of stupid. But we first discovered photosystems and photosystem 2 by looking at purple photosynthetic bacteria. So these are kind of interesting bacteria. They are photosynthetic. So they do conduct photosynthesis, and they have a lot of the same components of their photosystems as algae and plants. And so they're actually a great model system for studying the processes of photosynthesis. Now, photosystem 2 is a light harvesting complex. Okay, that means its main job is going to be harvesting light and transmitting that energy to do something else. So what's going to happen is we're going to have chlorophyll. We're going to have, remember this is a photosystem, we're going to have chlorophyll absorb those photons, use it to excite those electrons. They are going to be resonance energy transferred into that reaction center where we see a very specific set of pigments. Okay, pheophytin is going to be one of our main players. Okay, because what we're actually going to do with this electron is pass it down an electron transport chain. And yes, we have seen that before, right? Where have we seen electron transport chain? We've seen it in cellular respiration. And what does the electron transport chain do? We use it, right, to play a game of hot potato with an electron to pump protons into that inner membrane space of our mitochondria. And through that, we get ATP synthase, it spins, and because of this process, we end up creating ATP. So, if we are using this photosystem 2 to pass this electron through an electron transport chain, we can bet one of the main products of photosystem 2 is going to be ATP energy. So that's what our electron transport chain is, a game of hot potato, this time in our thylakoid instead of the mitochondria. Okay, we're going to see kind of structurally the same thing in our mitochondrial electron transport chain versus the thylakoid. So we're still going to see Q, right? Remember quinones, which we labeled Q, and we're going to see cytochromes. These are our shuttle proteins, so they're going to help transfer that electron between those members of our hot potato chain, and functionally very similar. Okay, we're pumping protons from one side of the membrane to another. We're creating that electrical gradient, electrochemical gradient, and we're using that proton motive force to make ATP using ATP synthase. So basically, structurally and functionally, this electron transport chain is the same one we see in cellular respiration. So here's kind of an overview of this. So we have our photon hitting our chlorophyll. It's exciting our electron, resonance energy transfer to that reaction center, exciting that electron, which gets given to pheophytin, and pheophytin's going to bring it over to the electron transport chain. We got PQ here, which is our shuttle, right? It's going to move it between the different members of our electron transport chain. And it's PQ here instead of Q because we're talking about photosynthesis. So this is technically plastoquinone, little different from our cellular respiration electron transport chain. But as that electron is passed to each player of that game of hot potato, ATP is going to be made through proton motive force, spinning of ATP synthase. So a few key players already, right? Plastoquinone, just a little different than our cellular respiration. We just knew it as quinone, Q, but PC is for photosynthesis. So let me draw it out for you. So very similar. We have our inner membrane, we have our outer membrane, but this little space instead of inner membrane space is called the lumen, if we're talking chloroplasts, remember, and this space out here on the inside is called the stroma. Stroma, just like that. So as we pass that electron from player to player, right, we're going to pump protons into that lumen, okay? Just like that. We're going to pump those protons in, and pretty soon we're going to get a high concentration of protons on the inside, right? Electrochemical gradients. We're going to get a ton of protons on the inside that all want to be moved back to the outside. They all want to go this direction. And we can use that force, that proton motive force, in order to generate ATP, right? ATP synthase, that lovely little molecule that sits, okay, right in that inner membrane. When the protons flow through it, ATP synthase spins and we create ATP. So again, exact same process we saw in cellular respiration. We're just in slightly different areas, and some of our players are a little bit different. Now, just like we said, protons diffuse down that gradient, going through ATP synthase, and we are going to see something called photophosphorylation. So this is a new one. We've seen substrate level phosphorylation. We've seen oxidative phosphorylation, but this one is new. Okay, and with this one, remember, what is phosphorylation? Addition of a phosphate group, so we're popping on a phosphate to ADP, right, adenosine diphosphate, in order to make ATP. But what's doing it is light energy. Okay, photophosphorylation. So that's what this process is. Photophosphorylation to create ATP in our chloroplasts. So here's that whole process all outlined for you. This is a really great diagram. It's showing all of this photosystem 2. You see we got the lumen here, stroma here, so it's a little bit flipped from my diagram that I showed you. We see light entering that light harvesting complex, resonance energy transfer, hits that reaction center, electron is given to pheophytin, which is given to plastoquinone, PQ, which shuttles it through our hot potato game. And each time it's shuttled, those protons get pumped in, and then those protons exit ATP synthase and create ATP. Okay, so don't reinvent the wheel here. It's the same process that we saw with our cellular respiration, just a few slight differences. Now, one thing that we have to ask ourselves is, how do we get that electron back? So if we think about it here, go back to this reaction center, right? That electron in our reaction center is excited and it's given to pheophytin, and then it's taken to its whole hot potato game, right? Now our reaction center does not have an electron anymore. That electron is gone. So if more light were to just come in, okay, it would have no electron to excite. So how do we replace that electron that we gave away to pheophytin? Well, that is where water comes in, okay, because we can actually split apart water, and we can use that to create some protons, some electrons, and some oxygen. This little equation right here is showing what happens when we split water, and we can use those electrons to replace that missing electron in our reaction center. And this is the step where oxygen is produced as a byproduct. So very important step, right? Because most of the oxygen that we breathe in comes from this oxygen produced by photosynthetic organisms. So this whole entire process is called oxygenic photosynthesis because we are releasing oxygen as a byproduct of our photosynthesis. Now, if we're talking organisms that only have a single photosystem, so they only have photosystem 2, they don't have photosystem 1, okay, they're actually not going to use water and they're not going to produce oxygen. So these organisms perform anoxygenic photosynthesis, so without oxygen photosynthesis. And so essentially they're going to use some other molecule to donate those electrons. An example of this, hydrogen sulfide in purple sulfur bacteria. So they'll release sulfur as a byproduct instead of oxygen. Okay, so that is photosystem 2, right? We are oxidizing water, we're splitting it apart, we're releasing oxygen, and we're using that electron to play a game of hot potato and produce ATP. So what about photosystem 1? Okay, that's our next photosystem we're going to talk about. And the key to photosystem 1, once again, we're going to be using light energy. We're going to harvest light. But what we are going to produce instead of ATP, we are going to produce high energy electron carriers. So our end goal is going to be NADPH, a full electron carrier. Okay, that's what we're going to produce with this next photosystem. So we are going to see photosystem 1 pass those excited electrons that are excited by light to that empty electron carrier to form the full electron carrier. So process is going to be somewhat similar. We're going to see the antenna pigments absorb the photons, resonance energy transfer to that reaction center. Electron in that reaction center is excited. Okay, that excited electron is given to a molecule called ferredoxin, and ferredoxin gives it to NADP plus in order to form NADPH. Okay, and we're going to see an enzyme that works with ferredoxin called NADP plus reductase. Okay, so it's just working with ferredoxin to hand that electron off to its final carrier. So here is what that looks like. Again, photon hits antenna pigments, resonance energy transfer, reaction center, electron is excited. Okay, that electron is given to ferredoxin, who gives it to NADP, right here, our empty electron carrier, to form NADPH, our full electron carrier. So now this molecule can go and it can donate that electron somewhere else. So, little summary. Photosystem 1 is all about producing high energy electron carriers, NADPH, and photosystem 2 is all about ATP synthesis. Okay, and this whole process of our photosystems is going to produce the chemical energy needed to drive our next set of reactions. So this energy that we made, ATP and NADPH, is going to be used in the Calvin cycle. Now, how are these photosystems connected? We call this connection the Z-scheme. Okay, how we kind of intertwine these two photosystems. And again, we start with photosystem 2 and move to photosystem 1. So, the key here is, how do we replace the electron in photosystem 1? Right? Think about it. Once we excite that electron and give it to ferredoxin, this reaction center is empty. But photosystem 1 doesn't split water. Okay, we can't use water to replenish it. So how do we replenish that electron? And that is the key fact of the Z-scheme. We can use the electron from photosystem 2 to replace the electron from photosystem 1. Okay, so let's see kind of how that looks. One key player we're going to have to keep in mind, this guy right here, plastocyanin. Okay, this is our connection molecule. This is what is responsible for the Z-scheme. Because remember, at the end of that electron transport chain, if we're talking cellular respiration, we would just give that electron to oxygen, okay, make some water, end of the day, we're done, right? But with this Z-scheme, we can take that empty electron at the end of the electron transport chain, and plastocyanin can shuttle it over to photosystem 1, and that is how we will replenish that electron. Okay, it is critical. It is the link. It is the connection. So let's look at this all together. Okay, here's our photosystem 2, which is what we start with. Photons excite, resonance energy reaches that reaction center. Okay, we're getting those electrons in the reaction center from splitting apart water. So water, oxygen's released as a byproduct. Electron gets given to pheophytin, then gets given to PQ, which passes it down that electron transport chain. We make some ATP. And then at the end of that chain, that used up empty electron is given to plastocyanin, which is able to shuttle it over to the reaction center of photosystem 1. Once we do that, use some energy, resonance energy transfer, we excite that electron, given to ferredoxin, and eventually given to a high energy electron carrier. Okay, so NADPH is what we create. So we used that electron from water and we gave it to our electron carrier at the end of the day. That is our flow of electrons throughout this process. And then it can start all over again, right? If we gave away the electrons in our photosystems, in our reaction centers, right? If we passed it on, okay, it's empty. Whole process can start again. We can split more water, give another electron, gets excited, passes all the way through. Okay, PC brings it to this reaction center, which is empty because we already excited that electron, so on and so forth. So this whole system, this Z scheme, and it kind of looks like a Z from the side if you follow it, like so. Okay, this Z scheme is the flow of electrons, and it is how these two systems are connected. So once again, this is showing you that exact same thing that the last diagram showed you, okay, with photosystem 2 and photosystem 1, and kind of just the whole process in a little more detail. I won't go through it again, but exact same thing we talked about in the last diagram. Now, we call this flow of electrons a non-cyclic electron flow, right? Because it's not a circle. We don't reuse those same electrons at the end of the day, right? They don't come back to photosystem 2. They progress in a nice straight line from photosystem 2 all the way to our electron carrier, which will take them somewhere else. So it's not a circle. They don't get reused. So non-cyclic flow, chain of redox, because remember, we're just passing an electron, which is redox, in a linear fashion. Okay, the terminal electron acceptor, so the thing at the very end of that line, okay, that is going to be NADP+. Okay, that's our terminal electron acceptor, because that is the last step that our electron will take before it is shuttled somewhere else. So just because we've described non-cyclic electron flow doesn't mean that there is such a thing as cyclic electron flow, so something that occurs in a circle. So, for example, some green algae and land plants will resort to the cyclic electron flow. And essentially with this, we're going to produce some ATP, but our photosystem 1 is going to act a little bit different. Okay, instead of using photosystem 1 to give that electron to ferredoxin, who gives it to our electron carrier, photosystem 1 is going to transfer that electron back to the electron transport chain. Okay, we will not produce any NADPH. Okay, we will not produce any of that. It's all about the electron transport chain. Now, the fun part about cyclic electron flow is that it actually can happen at the exact same time as non-cyclic electron flow. So all of this can happen all at once. So here's what's going on here. Here's our electron transport chain of photosystem 2. Here's our photosystem 1. Okay, our electrons get given to ferredoxin, but instead of becoming NADPH, right, they get given back to PQ, which takes it back to that game of hot potato, makes more ATP. Then that empty spent electron gets given to PC, who donates it back to that reaction center. So this whole process can start all over again. So this is our cyclic flow. Okay, our next set of reactions are going to be our light-independent reactions. So specifically, we're talking the Calvin cycle. We are going to take carbon dioxide and we are going to transform it into a carbohydrate, into a sugar. So to do that, we need to understand kind of some movement of carbon dioxide and oxygen. Right? So most plants are covered with this waxy cuticle, a coating. If you've ever gone and felt some leaves on a tree, you might have seen that they feel kind of waxy to the touch. And this is because this layer is a lipid layer that helps keep water in, right? Because we need water for this entire process. That's what we need for our photosystem 2. And it's also going to have the added effect, which is not necessarily a good effect, but it's going to prevent carbon dioxide and oxygen from moving in and out of our cells, okay, of our plant cells. Now, this is kind of bad, right? We do need to keep water in. But if we don't import CO2, there will be nothing to make sugar out of, okay? No carbohydrate. And if we have too much oxygen that we produce that has no way of getting released, that's also going to be bad. So we have to figure out a way to capture and bring carbon dioxide into our plant tissues. And this happens through pores. Okay, these pores are little openings on the surface of our cells, our leaf cells. They have two little guard cells on the side. And this whole unit, so the two guard cells and the pore, is called a stoma. You see multiple together, you call that stomata. So anytime you may have watered a plant, you may have noticed this as well, water kind of beading up on the surface. And this is because of that waxy cuticle, that lipid layer. Now, when these stomata are open, that means CO2 can come in and oxygen can go out. And that's what we want. And this movement is going to happen because, once again, those concentration gradients, right? If we have more carbon dioxide on the outside of our leaf than on the inside, when we open that doorway, carbon dioxide is going to move in. It's going to move out of that crowded party room and into the chloroplast. Same thing with oxygen. Now, we always want a good amount of CO2 in our plant cells because, again, this is what the Calvin cycle is going to use to make sugar. We're going to fix carbon in order to make a sugar. So here are those stomata we can see on the leaf surface, guard cells on either side, and the pore opening. Same thing down here, but in a diagram. We got some guard cells and we got the pore. Okay, and you can see in this one we have some carbon dioxide moving in and some water and oxygen moving out. So the first step of our Calvin cycle is called carbon fixation. Essentially, we have to process carbon dioxide. It can't be used in the Calvin cycle immediately. We got to process it a little bit first. And it was Melvin Calvin who ran the first experiments to kind of figure this out, hence the Calvin cycle. And he noticed that it was a 5-carbon compound called ribulose biphosphate. Okay, that is going to be the initial reactant. Okay, and this equation, this chemical reaction, this fixation of CO2 to RUBP, which is that ribulose biphosphate, the fixed carbon that we can use, is one of the most important chemical reactions on Earth. Okay, this is what is responsible for the creation of that carbohydrate from light energy, which fuels so many different food webs on Earth. So, what does the fixation? What transforms CO2 into RUBP? And the answer is an enzyme called Rubisco. Okay, specifically ribulose 1,5-biphosphate carboxylase oxygenase. Whew, really a mouthful, which is why we call it Rubisco. Now, this is one of our most abundant enzymes because it's, once again, it's what's going to fix our carbon. It's what's going to fix it into RUBP, something that we can use. Now, here is RUBP, very large enzyme, 8 different active sites, so it can process a lot of stuff at once. But the thing about Rubisco is that it kind of sucks at its job. Okay, it is not very good at fixing carbon dioxide. It's slow. It's inefficient. And what we mean by inefficient is that it can't really tell the difference between carbon dioxide or oxygen. Okay, it'll use either one. So, really not good at its job. And we actually do not want oxygen fixed to RUBP. And we're going to see why. But if we're fixing oxygen, right, we now have slowed the rate of CO2 fixation, right? If we have oxygen and CO2 kind of competing for these enzyme active sites. Now, oxygen and RUBP, when they react with Rubisco, we call this process photolysis. respiration, light and respiration. Now, the reason that this is so bad is because photorespiration basically undoes photosynthesis. Okay, instead of producing oxygen, it's going to consume it. And what it produces is CO2. Okay, so consume oxygen, produce fixed CO2. That's not what we want. We want to release oxygen and we want to produce a carbohydrate at the end of the day. So photorespiration is bad. We want to minimize photorespiration as much as possible. So here are those different reactions, right? We have RUBP, we have carbon dioxide with Rubisco to make some sort of product that's used in the Calvin cycle. This is what we love, okay? This is photosynthesis. Great, lovely, plants love this. Okay, if we look at photorespiration, RUBP, using oxygen with Rubisco, and we do get something that's used in the Calvin cycle, but we get something else where that releases CO2 and uses ATP, which is, again, not what we want. We do not want this. This is not a good reaction for our plants. So how can we minimize photorespiration? Are there any ways that we can ensure that our plant does not resort to photorespiration? Now, if conditions are kind of normal, so wet and kind of neutral temperatures, right? That's not really a problem. We can open those stomata, carbon dioxide can come in, oxygen can go out, no issues, okay? Photosynthesis can proceed. However, in hot and dry conditions, we have to close those stomata, right? We have to stop. We have to hoard all the water we can if it's really hot and dry outside. But whenever we close those pores, CO2 and oxygen are not going to be transported. So we have less CO2 and we have more oxygen. Okay, this is bad because if we close those stomata, we're going to see an increase in photorespiration. So photosynthesis slows, photorespiration increases. This is bad. So how can we fix this? Okay, and we're going to see kind of two fixes to this process called C4 and CAM. Okay, two pathways that we can use to essentially hoard carbon dioxide. Okay, that's our goal. Now, the other pathway we're going to see is called the C3 pathway. Okay, and this is just our lovely little standard pathway, which is where we're taking RUBP plus carbon dioxide into some sort of product. So this is standard. This is our standard carbon fixation. Okay, so I've drawn out our little C3 pathway. So again, this is using RUBP plus CO2. We're using our really inefficient enzyme Rubisco, and we get some sort of fixed carbon product. So this is something that's going to go into the Calvin cycle, the next step of the Calvin cycle. Okay, so this is our classic. This is what's happening, okay? But when things get hot and dry, that's where we have to make some modifications. So we're going to kind of use this pathway still, but we're going to do some extra things, specifically C4 and CAM. So kind of two extra pathways that we'll use in order to kind of pump carbon dioxide back into this C3 pathway. Okay, so that's kind of how we're breaking this down. So keep in mind our C3 pathway, because we're going to see it come back into play when I discuss the next two pathways. So we're going to start with C4, and C4 plants, okay, fix carbon dioxide using both pathways. So what that means is we are going to use C3, our standard, okay, that's always going to be running. But we're going to do a little something extra, okay, and that's our C4 pathway here. So C3 is still running. We still see this whole thing going on. But now we're going to do a little something extra, okay, and that little something extra is this C4 pathway where we are going to essentially pump carbon dioxide. So we are going to use an enzyme called PEP carboxylase, and this one's new to the C4 pathway. And it's essentially going to funnel in more carbon dioxide into this C3 pathway. And I want to emphasize, right, we're not replacing the Calvin cycle. This is just an additional fixation step. So we're doing C3 plus C4. Okay, just additional step in hot and dry conditions. Now, the key for the C4 pathway that you will absolutely need to remember. Okay, C4 pathways are spatially separated. Okay, our C3 and C4 pathways are found in distinct cell types. Okay, we're going to see PEP carboxylase, so our C4 is going to occur in something called mesophyll cells, and our C3 is going to occur in bundle sheath cells. Now, you don't have to memorize what type of cells, but know that they are happening in different geographic locations within our plant. So let's see what that looks like. Here is our C4 cycle taking in some carbon dioxide, and you'll see that as this cycle progresses, right, it's going to funnel some CO2 into our C3 pathway, right, our Rubisco carbon fixation pathway. But the key, look at the cells that it's happening in. It is happening in two distinct cell types. So we call that, once again, spatially separated. But that's what the C4 cycle is doing. It is a CO2 pump. It is pumping carbon dioxide into this C3 pathway, which is part of our Calvin cycle. Now, our next carbon pump is called the CAM pathway. Crassulacean acid metabolism, CAM pathway. Now, once again, it's a photosynthetic pathway. It's going to be a CO2 pump, okay, and it's going to look a whole heck of a lot like our C4 pathway. So we're going to have a set of C4 reactions, okay, and our C3 reactions are still going to occur. So this is still a C3 plus C4, okay. The main difference with this is CAM plants are not going to spatially separate these reactions. Okay, so these are going to happen in the same place, but the key, different times. And CAM plants are going to be things like cacti, really desert adapted species that have to keep their stomata completely closed on any hot, dry days. So, big difference here. Okay, our C4 cycle, our carbon pump, is running at night. Okay, so when it's not quite so hot and dry, we can open up those stomata, we can run this C4 cycle, we can concentrate a bunch of CO2, and then during the day, we can funnel in that CO2 that we just made into this C3 pathway, this carbon fixation that we do as our first step of the Calvin cycle. So that's our main difference. They're both CO2 pumps. Okay, they're both for hot and dry conditions, so when we need to keep those stomata closed. But if we're talking C4, spatially separate, right, we keep them in different cells. And CAM plants temporally separate. Okay, we do these cycles at different times. Okay, that's our main differences here. So these are very important kind of key points that differentiate the two. So that was our first phase of the Calvin cycle. Check. Okay, we described fixation. So CO2 reacts with RUBP, and we're going to make some sort of fixed carbon product. And that product, we can go ahead and give it a name now, is PGA. Okay, that is our fixed carbon product, which now can move into our next major phase of the Calvin cycle, the reduction phase. And the reduction phase is where we take that PGA that we just made, and this is where we're going to use our ATP and NADPH, that full electron carrier, from our light-dependent reaction. So this is where what we just produced in photosystem 1 and 2 are going to come into play. Okay, reduction. And then our last step is called regeneration. So remember, this is the Calvin cycle after all, so it is going to be in a cycle, in a circle. And our product from reduction, okay, some of it's going to be used to make carbohydrates, and the rest is going to be used to regenerate our UBP. Okay, so we can start this whole process over again. So, 3-step process. All of this is going to take place in the stroma, so inside that kind of fluid of the chloroplast. And one turn of this cycle that we just described is going to fix 1 molecule of CO2. So what does that all look like together? Well, here is our Calvin cycle, the 3 phases. Right, we've already seen fixation. Okay, we're using RUBP, CO2 to make PGA. Reduction, that's where we take our PGA that we just made, and this is where we use ATP and NADPH, and we get a product, G3P. Now, we get 1 net G3P that's going to go on and make a carbohydrate, and the rest is going to be used in our regeneration. So we're going to use it with a little bit of energy to remake our RUBP, which now can be reused into our first step of fixation again. Okay, and this is all of it in a cycle. So same exact thing. You can see where we're using ATP and NADPH. You can see our product that is going to go and be used to make a carbohydrate, and the regeneration step where we remake that RUBP. So what about regulation? How do we regulate photosynthesis? What if we have enough carbohydrates? Right, so there's a few different ways that we can stop and start this process. Now, one of them is the presence of light, right? If we don't have any light energy, photosynthesis cannot occur. So if we have light, that's going to trigger the production of a lot of proteins that are responsible for these reactions that we see. We also see high sugar, right? If we have enough carbohydrates, we don't need to make more. So having a lot of sugar, a lot of carbs, can kind of stop the production of these proteins, these photosynthetic proteins that make more carbs. And it's actually going to stimulate the production of other proteins that are going to start converting that carb that we just made into things like starch to store that sugar away for a rainy day. Now, Rubisco, our enzyme that's also pretty terrible at its job, can also be regulated, again, by light. So if light is available, Rubisco will be active. However, when CO2 is low, Rubisco is going to be inhibited, and instead we're going to see, again, photorespiration be preferred. Now, what happens to that G3P, right? That was our yield of the Calvin cycle, and this G3P isn't quite a carbohydrate yet. It's just the precursor. Okay, so it's the molecule that it's going to be made into a carbohydrate. So that G3P can either make glucose, it can make fructose, it can even be combined together to make sucrose, a more complex sugar. And whenever we are making a carbohydrate, we call that gluconeogenesis. Genesis meaning creation, and glucose referring to a sugar. So again, if we have a lot of sucrose, right, we can kind of stop the process of photosynthesis, and instead we can start promoting proteins that will polymerize that glucose, the monomer, into starch, so a storage carbohydrate. Once again, production of starch is all going to happen in the chloroplast, but if we're talking sucrose synthesis, we want to make a more complex sugar, that takes place in the cytosol.